Thank you, Tanya, for that introduction. You're always very generous and kind. Hello, everybody. We're getting to the end. We got 2 more talks to go. I know everybody must be really tired by now, but I promise I have something really exciting. I'm going to talk about jeans, not the ones that I'm wearing, but the ones that make you, you. I'm Shweta, and I'm a principal engineer at Recursion. Recursion is a clinical stage tech bio drug discovery company. Scientists at Recursion are focused on using technologies to identify novel medicines to treat diseases. Our approach at Recursion relies on building massive proprietary data sets of biology and chemistry that can predict trillions of relationships across biology and chemistry, many of which are not known in scientific literature. We do this by running millions of experiments in our lab every week. One such type of experiments are CRISPR-Cas9 mediated knockout experiments. In these experiments, we knock out every gene in a human genome and understand the activity level of each gene for a specific disease model. This is an example of a gene knocked out in a cell. In our labs, these are robots that execute these experiments. They take images of the cells, as you can see in that picture. And these images are fed through a deep learning model to generate features or deep learning embeddings. These embeddings are used by data scientists to create custom computational methods to build maps of biology and chemistry. Every data point on that map represents a relationship between genes. Red indicates similarity, blue, the negative or opposite relationship. These maps can be used to wade through areas of known biology and thus allow us to explore the unknown, which prior to building these maps was not very trivial. These experiments run every week in our lab, and they generate novel insights and relationships that we periodically need to add to these maps. Each map that you can see here, this is a whole map, is built for a specific cell type. We knew how to build a map for a cell type by the end of 2021. But grand challenges for Recursion in 2022 involved building an atlas. An atlas is a collection of maps. This talk is about how we were able to scale our map building process to enable the build out of this atlas. So how did we build maps at Recursion circa 2021? To put this talk together, it felt like opening a time capsule. A lot of things have changed since then. Scientists request map building experiments to be run in the lab. These experiments take time to execute. They would periodically check with the product manager if a new map version included their experiments and if their experiments were done executing. The product manager would scroll through several Slack channels to find those experiments, and she would keep a running list of experiments for that week. And then she would Slack those experiments to data scientists and engineers to build a new version of the map. This wasn't when engineers made time to build this new map or new version of the map, they ran a set of scripts to build this map. This wasn't a trivial exercise because sometimes these scripts would fail, processes time out, and it took several attempts before the new experiments got added to the map. This process was very cumbersome, inefficient, but it was a process that built maps at Recursion. Recursion championed this concept of maps of biology to discover novel insights around mid-2021. This process, however rudimentary, allowed us to validate that we could use these maps to verify known biology and hence could use them to explore the uncharted areas of biology. At this point, I want to share a directive in software, Ken Beck's directive. I don't know how many people know of Ken Beck, but he's a software engineer and a creator of extreme programming. I'm not here trying to champion extreme programming, but this one directive is something that I really like and keep in mind when building systems. Make it work, make it right, and then make it fast. Using this analogy, we had made maps work. We had made them right. And now, with the grand challenges in 2022 looming ahead of us, we had to make them fast. Not only did we want to build one map, we wanted to build an atlas. One way to make things fast is you want to figure out how to take these scripts, run them automatically, or create an infrastructure to run them. Fix the individual pieces, build the map faster. But in this case, the entire story, the big picture, was important. I had to get people outside the tech org involved to solve this problem. We had the experiment operations team, we had the scientists, and more importantly, the product manager. I had to bring systems thinking to this problem. To start with, we wanted to identify the map building experiments. Remember, the product manager would scroll through these Slack channels. I talked to the experiment operations team, the product manager, and we thought about something. If we tagged these experiments at design time, we could obtain this list programmatically. I knew that these tags were stored in the database, so that scrolling of Slack channels could be replaced by a SQL query. Nothing fancy here, but knowing the big picture, how the experiment design team operates, was important to come together and create this solution. No more Slack scrolling for the product manager. During this process, I also uncovered a piece of information. Not all the experiments executed went into the map. They needed to pass QC, or quality control. One way to solve this was to build an event-driven system, one that would emit events that when the experiments are tagged, or when the map building process, and then the map building process would be notified, and that would really solve all the problems. This solution, however, required more than one engineering team, several operational teams. Making that work wasn't trivial given the time constraints and the priorities that other teams and ours had. So it seemed best to maybe establish an SLA with the QC team. Working closely with product and operations team, we decided experiments that are QC'd by 5 p.m., make it into the map. Very simple. So we had a list of experiments obtained programmatically. I talked to the data scientists and engineers. If we had this list now, would we be able to automate the process of making maps? We took the scripts, the data scientists and engineers we were able to put together an architecture, a cron job that would run these scripts and like my product manager says, would do the thing. It would add the experiments into the map. We added Slack integration to this automation. Every Monday, once the cron job ran, built a new version of the map, we told the scientists were notified. They knew all the experiments that were in that map, and they could scroll and see without bugging the product manager. They were happy to see they knew exactly where to go to know whether their experiment made it into the map. When you put together a system like this, an end-to-end system to build a map for a specific cell type, building multiple maps is trivial. Cell types can be looked upon as a function argument at that point, instead of reinventing the wheel to build a new system for a different cell type. To be able to see this and lead teams to enable priorities for recursion is something that brings me immense joy and satisfaction. What I shared today is more than just building maps of biology and chemistry. You can use these principles and apply them to your domain. Understand the system you're building. Understand your users. Use systems thinking to solve problems. Think end-to-end than just one piece. Start small, make things work, make them better, and then make them fast. Find ways to deliver product value iteratively. Thank you, Lead Dev and StaffPlus, for giving me this opportunity to share this story. And I hope you enjoyed the last talk.